Hi, everyone. Thank you so much for coming to Wednesday evening at the Genome. It's a real pleasure for me to welcome Dr. Rosana Risques. So she's a professor in the Department of Pathology that's just across the street from Genome Sciences. And she's a real pioneer in a technology called duplex sequencing, which is this super accurate type of DNA sequencing where you sequence the whole DNA molecule and not just half of it. So Dr. Risques got her BS and her PhD at the Autonomous University of Barcelona. And then she went on to postdoctoral training at the Sidney Kimmel Cancer Center in San Diego before doing her postdoc right here at University of Washington, and then moving up through the professor ranks. And her research interests involve somatic mutations in the tissues of the body, and how they can progress to cancer and how studying them can inform the early detection of different types of cancers. So with that, I'll turn it over to Dr. Risques.
Thank you, Kelly, and thank you for the opportunity to speak here. This is always a privilege. And thank you for all of you that are here with this beautiful sunny afternoon or evening. And I think this is great, even if it's just a few of us, but I hope you are here to learn, and I think you'll learn a lot. And the first thing I'm going to do is to ask, how many of you do not have a background in genetics? Just a few. Okay, that's great. So thank you for being here. That's part of what I was taught, is that this is public speaking. People are not assumed to know genetics. And so I've put our research here in a very plain, easy way to understand, I hope, so that you can learn from it. And this will be interactive. We are few, and this is supposed to be fun and learning. So all questions are welcome. So the first thing that I wanted to say, I just mentioned briefly how fortunate I feel to do this, because if you had told me when I was just here, this is Barcelona, Spain, my home city, doing a PhD many years ago, that one day I'll be speaking here in one of the best genome sciences departments in the States and surrounded by these stellar speakers, I would not have believed that ever. And I want to tell you, though, that this was not as beautiful as this straight pathway. It was more like this. So I came from a disadvantaged background, and it was not linear. It was not easy. I didn't see my pathway. But I'm showing you this because some of you might be thinking about being scientists and might be thinking, do I fit in this? And the answer is yes, you do. If you have passion, if you want to do this, you do fit. So follow your passion and it will lead you. So then after that, I'm just going to tell you science. And so what we are going to talk is about cancer. Cancer is an evolutionary process. So this is what I'm going to explain first. Then our work in ovarian cancer detection. And then we're going to talk a little bit about the colon if we have time at the end. But before doing that, I do have some disclosures because I am a consultant and I have equity in a company, this company here. We have patents also owned by the university that were licensed to this company. And we have had some research grants. And I also have equity in this other company. So this has to be said. And now we can jump into the science. So the research in my lab focuses on how do cancers develop. And we are interested in this for two reasons. One is biological. I'm a biologist by training, and I want to understand. We want to know what is going on. The second reason is prevention and early detection. And this is obviously very important. We think that the best way to treat cancer is not to get cancer to start with. So we want to work on this very, very intensely. And so my lab focuses in this part here. This is a cancer, this is earlier. And what we want to know is how a cell here in green eventually becomes this. And what we know is that this happens with mutations. So I'm going to give you a super brief introduction about mutations. So mutations are changes in the bases of the DNA or nucleotides. And I'm not going to explain everything to you. I'm just going to say that sometimes the changes are okay, like these ones that are synonymous, that's fine. Many times they are really bad because they make the protein not to be produced. This is a stop for production of the protein. So this is bad. And these missense, non-conservative, they change the amino acid, they change the protein enough that it won't function, that's bad. Insertions and deletions, we call indels, those are really bad too. We call this pathogenic. So mutations that are bad in the context of cancer, that they might lead to a cancer. So we know that cancer is an uncontrolled cell growth driven by mutations. We need many of them. And so this cell gets mutations and so on. There are more things that happen. This is a bit simplistic, but mutations are essential. Now, what is not that clear is this linear model that I put there is not that linear, right? So this is more complex. What happens, in fact, is evolution. Evolution as described by Darwin. Mutations, I'm going to put it here. Mutation, selection, and clonal expansion. I'm going to explain this in a second. So if we get a mutation here in a cell, if this mutation gives some advantage to the cell might proliferate, right? So that makes a clone, is a clonal expansion. This mutation is being selected because it gives a feature to the cell that allows it to grow, right? So this happens multiple times in cancer, and that's the process of evolution. This was described by this person, Peter Nowell, in 1976, before we even were sequencing DNA at all. So for me, it's very remarkable, and I always put this slide because it's just amazing how he envisioned cancer as an evolutionary process, and this is indeed what it is. And so what happens, again, to clarify this idea, which I know is complex, is that these cells get pathogenic mutations and they expand. I'm going to be using in the talk the term clonal expansion a lot. So that's what I mean. I mean a clone of cells that carry a pathogenic mutation, and this keeps happening. So for this to happen, then we are hearing two things. We need mutations, and we also need time. For mutations, actually, we have plenty. So what we know is that mutations are inevitable. So if you know DNA a bit and you know cell replications, when the cells have to replicate, they copy their DNA. That's the replication of DNA produces mutations. It's not perfect. We also have the environment where the cells live in our body. That also leads to mutations many times, the microbiome, inflammation. And in addition, we have mutations that come from the outside, smoking, diet, UV light, all this creates mutations. We estimate that maybe as many as 1 to 6 mutations per cell division. And in terms of time, we are humans, we live long, and so we have years to accrue these mutations in our cells over time. Fortunately, this doesn't happen to everybody, but mutations happen, and cancer is a disease of aging, right? So with the exception of childhood cancers, which is a different story, most adult cancers happen late in life. We need the time. Of course, if you do things like this, or like that, then you are going to accelerate this process of getting mutations. And I want to use this slide to remind you that there are also some cancers that are hereditary, not many, maybe 15%, 20%. And what happens is that people inherit one mutated allele, one copy of one gene comes already mutated. So this accelerates this process for these people, yes? Okay, so if we want to study cancer, we have to measure mutations. We have to detect them. How do we do that? So this is the old-fashioned way to do it. And I like to put this slide with a bit of nostalgia because I'm that old. And so I did sequence like this with radioactivity back in the day in my PhD time. And so that's kind of fun to say that I did that. And of course, this is a long time ago. Very low throughput, very slow. You are reading the letters of the DNA. You see here the DNA code one by one. It can be done with fluorescence, but in general, the problem here is that you have to have the mutation almost in all the cells. This has no further resolution. Now, since 2000 or so, we have what has revolutionized cancer research and medicine and genetics, which is what we call next generation sequencing. This type of sequencing is digital, is much more high throughput. Most importantly, it is digital. So what we have here are reads, and I'm not going to go into detail here, but these are pieces of DNA. What we do is align them against the reference genome here. And so we see there are changes. So here you can see a C in the reference genome that now has mutated to a T. In this case, all the reads carry this mutation. That's what we would say is a variant allele fraction, a percent of 100%. But here with this method, we can go as low as 2%, just a few reads, 2% carry the mutation, we can see it. So this is obviously very powerful and has been used a lot. So what we've done with next-gen sequencing in the field of cancer is sequencing tumors, right? So there was this Cancer Genome Atlas initiative. I'm not gonna go in detail, but you can see some of the numbers here. 33 different tumor types, 11,000 patients, so a lot of sequencing data here. And there are other international efforts. All this data is available. We call it COSMIC, Catalog of Somatic Mutations in Cancer. So definitely a lot of knowledge has been generated about what mutations are in tumors. Part of the knowledge, going back to evolution, is to infer how tumors were produced. So this is one of my favorite papers with many cancers studied in detail. You don't have to understand any of this. The point here is to show how complex it is. And because of the tumors having so many mutations, doing some computational tricks, you can figure out when they happened. And so the prediction is that they happen very early in life, many of these mutations. And so the question for us is still, okay, that's amazing to infer, but this is still tumors. Can we see early cancer? And can we study this better than just sequencing the tumors? And the answer is yes, and I'm going to show you how. So first, just to clarify, this is the tumor. Next-gen sequencing detects mutations at this frequency. We want to be here. So to be here, what we need is high-resolution sequencing. So we want to be able to detect one mutant cell or a few in 1,000 or more of other cells that are there that are totally normal, right? And so this is like finding a needle in a haystack. You need that sort of resolution. And we also need that when we do something that we call liquid biopsies. So when we try to get biopsies from patients and try to find the tumor mutations there, that's also very hard to do and you need high resolution. And so the good news is that now we have a method to do this. That's what Kelly was referring earlier. It's called duplex sequencing. UW Proud, this was invented here just across the street from the prior pathology department. And our colleagues had the brilliant idea to think that DNA is double-stranded, right? So DNA has two strands. The copy, the information is copied, is double. And so to use that, what they thought we could do was to put barcodes to the DNA molecules and then go into sequencing. We produced these reads that I showed you before. The idea here is that we can group all the reads that come from one DNA molecule, one strand, and then the other strand as well. And when we call the sequence, then we call a consensus sequence. And that removes errors that might happen during the whole process of sequencing, which is actually very error prone. And when we compare the two strands of DNA, the mutation has to be in the two of them. So it's kind of an extra level of double checking, right? So this removes a lot of errors and it gives us an error rate less than 1 in 10 billion. So it's very million in 10 million. It's very high resolution. So the summary of all this part is that cancers form through mutation, selection, and clonal expansion. We call this somatic evolution. Cancers require multiple mutations and many years to form, and that's why it is a disease of aging. We know a lot about what happens here, thanks to this next generation sequencing that I just told you about, but we don't know much about the early stages of cancer. However, we have now high resolution sequencing, so we hope we can start understanding what is going on here. So I'm going to take a break here because I know I've packed a lot very quickly in a bunch of slides. Do you have any questions? Probably many questions, but any question that I can answer now for you? Colleen.
If there's a patient who presents with a tumor that is undeveloped, is there a way to use sequencing to detect like the tumor cells in a different part of the body so that you can find where the primary tumor site was found?
Yeah, so Colleen is referring to the site of origin or the tissue of origin of some cancers, right? So you might not know sometimes when a patient comes to the clinic if that tumor that is showing in the liver is a liver tumor or is coming from the colon, because these cells tend to go to the liver. That's called metastasis. With our method of sequencing, and any sequencing for that matter, you don't need very high resolution there, but what you can do is trace the mutations. The mutations, as cells divide, get inherited. So it's kind of a fingerprint. And so you can build the whole evolutionary tree of tumors and then try to see where things come from. That's assuming that you have a biopsy of the colon, the liver, the breast, wherever you have metastasis, you can sequence all that and then try to infer this evolution with that. Yeah. Questions?
What was the benefit of using next-generation sequencing from other forms of sequencing?
Yeah, so the main advantage, as I said, is that it's high throughput, right? So you can sequence many more samples, a lot of expansion of DNA, not just one gene, but all the whole genome. And it's actually digital. So you don't need all the cells to have the mutation. You can have only 2% of the cells with the mutation, and you'll detect the mutation there. And if you use duplex sequencing, you can have only one mutant cell in 2,000, 5,000, and still find the mutation. So that's the high sensitivity that this method has. That's what we want to use in here. We want to find only one cell that is mutated. The rest are all normal. Yeah, great questions. Okay, one more, yeah.
Available is like sequencing. You need a special machine, with the equipment and type. Is it like you can buy a kit and do it in your lab?
Yeah, you can buy a kit, so it's commercially available. And the machine and the whole process, fortunately, is very similar to standard next-gen sequencing. So that's what everybody does these days. There are other methods, but there is a big company that produces the sequencers and the kits. This is just a variant of that. So it doesn't need a specialized equipment. The analysis is tricky, though. It's a little bit different. So you have to learn how to analyze this sort of data. But for the most part, it's just getting the kits. You can also do it yourself. So potentially it can be done in the lab, but that's how we did it back in the day. But it's not that easy. Always easier to buy if it's available. Yeah, great. Okay, so now I'm going to show you what we are doing with these new methods and what we are finding, which has been really unexpected. So the first thing I'm going to explain is why did we focus on ovarian cancer? And the reason is because this is a cancer type that gets detected at a very late stage. And that means that the survival is very low, is 25% only. And the problem is that there is no early detection test. And so that's why most women don't recognize the symptoms. By the time they go to the doctor, it's too late. Now, biologically, something that is a bit unique with this cancer type is that it has one gene that drives most of these cancers. We call them drivers. And this gene is called TP53. It's mutated in almost all of the high-grade serous ovarian cancers. This is the cancer type that we work with. It's the one that is the most common and the most malignant. And this gene here, so that you don't get confused, the gene makes a protein that is called P53. So it gets a bit confusing here, but that's the gene, that's the protein. I'm going to show you here pictures of the protein. And what is important is not only that most of the cancers carry this mutation, this gene is very commonly mutated in other cancer types, by the way, as well. But typically other cancer types have other genes mutated. In here is pretty exclusive, this one, and others that happen later. But this is the initial one, and that's what is important. It's present in very early lesions in the fallopian tube. And so that's a really good target if we want to do early cancer detection. So now you can see here the fallopian tube epithelium. And I have to explain that even though this is ovarian cancer, it originates in the fallopian tube epithelium. So not to be confused here, that's the biology. These cells start here and then they go to the ovary. And at the beginning, the epithelium is normal. And when you see this brown color, is that there is a lot of P53 protein, and that's abnormal. And so you see then these cells just crowding more and growing more, and this is now a full carcinoma. So what we thought we could do is two different approaches to try to improve ovarian cancer detection. One is called uterine lavage, and the other one is called pap test. The pap test is the typical pap test that is used in the clinic to detect cervical cancer. The uterine lavage is not typically used, but we use it because our colleagues invented this catheter that what it does is put 10 mils of saline solution into the uterus. It rinses quickly and collects the liquid. The idea is that if there is an ovarian cancer, these cells disseminate a lot, even very early. They move out of the fallopian tube, and then we think they might come in here and we can get them. And so we can see then during this test if an individual has ovarian cancer or not. So that's what we tried. And this is the biggest slide with data. So I'll explain little by little. So what we did was a very small pilot study with 10 patients with ovarian cancer and 10 patients without cancer. What I'm showing you here is the sequencing data for TP53, this gene, in these patients from the uterine lavage. If you see red here, these red columns, those are the tumor TP53 mutations. We knew what was the mutation, the specific one in these tumors, and we found them in the lavages. So this is great results. That means that this is the number, 8 out of 10, 80% sensitivity of finding the tumor mutation means that we have potentially a very good assay to use in the clinic because this lavage is detecting a tumor mutation. We know these cells that we got here came from here. Now, the big surprise is that you see other blue bar here. These are other TP53 mutations. And that we did not expect. And we saw many TP53 mutations in people with ovarian cancer and in people without ovarian cancer. I told you this is a cancer gene. So what are these mutations doing here? So that was our big question. Is this noise? This method is probably not that good because we are seeing a lot of stuff here that is not supposed to be there. Now, the surprise was that when we decided to count these mutations and then do what we call a mutation frequency per patient, and then we plotted by age, it correlated with age. The older the patients, the more mutations. So that gave us a hint that this is biological. This is not error. And then the other big clue here was that if we get these mutations and we plot them in the gene, this is the TP53 gene, this middle part of the gene is actually where all the action is. So the bad mutations, the pathogenic mutations typically occur here. And that's where we saw our mutations clustering. And so to make this clearer, we compared with mutations found in tumors. I told you before we had sequenced many tumors, not we, but the community. And look at this, it's a mirror image. So that means that the mutations we are finding in this lavage are very similar to the mutations that are in cancers. And this was very surprising. What this means that these mutations are not random. If this was noise, this would be all over the place. We wouldn't have any pattern here. But these are pathogenic mutations found in individuals with and without cancer. We found these in both. So then the question here was, really, how could it be? Do we really all carry TP53 mutations inside of us? I mean, I don't know all, but all the people in our study had. And we were surprised, but not that much, because if you start looking at the literature, some other groups have seen similar things, right? So this was the first study here. I'll explain in a second. This is from my group and this other study as well. And so others were coming to with this same sort of discovery, that there are cancer mutations in individuals or in tissues without cancer. And so with my colleague, Scott Kennedy, and I, we wrote this review where we said, well, this is like an iceberg, right? So back in the day, we had not much sensitivity to find mutations, and we just saw a few in normal tissue. People had described that. Now that we have a lot of sensitivity, we find all these mutations. And what we wanted to know is where are they coming from, right? Because we're just sequencing this lavage, and this lavage is collecting what? And so what we did was just sampling different tissues. We got fortunate we could access tissue from a newborn to a centenarian. And this was not the best designed study, but we had multiple different gynecological tissues. and we checked for TP53 mutations. And what we found is that the ones that are not common in cancer, they were kind of all over here. Might not be errors of sequencing, but the important ones, the common in cancer, were not in the newborn, and then they increase with age. And so this told us that these mutations, indeed, the ones that are pathogenic, are not early in life, but they accumulate later in life. And Colleen, who is in here, a grad student in my lab, did a much better design study using normal fallopian tube, again, a wide range of ages from newborn to 91 years of age. And what we've done now is sequence very well and very deep TP53, and you can see that there are many more mutations here. So indeed, they accumulate with age, and the color here in orange are more pathogenic mutations. So you can see there is more orange here, and darker colors mean also larger clones, right? These clonal expansions that I told you. So what this is showing is that large clonal expansions occur in individuals without cancer. This happens with aging. And now what we want to figure out is, well, do individuals that are at risk of cancer, like those that carry BRCA1 and BRCA2 mutations, you might have heard of that. This is the story of Angelina Jolie that was popular years ago and really benefit the field to know that there are people that inherit these mutations and they are predisposed to breast and ovarian cancer. So now in my lab, we are studying these individuals, their fallopian tubes, to see if their high risk of cancer is related to more of these mutations. It might not be. We know this is normal aging, but it could be that in some patients, this is accelerated. They are getting more of these mutant clones, and that predisposes to developing cancer. So that's part of what we are doing. And before I continue, I want to take just a brief detour to tell you what has happened in this field of clonal expansions, right? So I told you from my lab, but this has been growing a lot in the last decade. And the most famous tissue that has been analyzed is actually blood. And in blood, these mutations have been identified, and they are called clonal hematopoiesis of indeterminate potential. So big name, it's abbreviated as CHIP. And these CHIP mutations are detected in the elderly, and they predispose to leukemias, but also to cardiovascular disease. This is one of these mysteries that is still being investigated, but this is a very hot topic now, understanding these clonal expansions in the blood. In solid tissue, I told you a few slides ago, this paper in 2015 was the first one that saw this. So this study was analyzing skin in the eyelids. So you have here a beautiful picture. This is obviously not at a scale. These are microscopic clones that are drawn based on the data. And so what this study found is that there were all these genes that are cancer genes mutated in the skin of individuals that do not have cancer in the eyelids. And so that was the first big paper that opened the field, essentially. The same group did an analysis of normal esophagus in young, middle-aged, and old, and also used the same color coding to show how the clones increase with aging. And again, cancer mutations in most of the cases. And so what I tried to do at some point was summarize all the data, and I started this in 2019. So many more papers coming, finding these cancer-associated mutations in normal tissues. The studies here were the ones done just incidental, we call by chance. We found these mutations, we were not looking for them. And then more studies have come later on, analyzing all types of human tissues, trying to find these cancer mutations there, and they are everywhere. This is normal aging. We know now this is the new normal. Okay, and so I stopped doing my review there because now there are excellent reviews in the literature. So this is the best one in 2021 that it explains in detail all these new studies with all these cancer mutations in normal tissue. And the most important part here is that it goes in detail into what could be causing this. So we know it's aging, but it could also be inflammation. It could be the environment. It could be genetic predisposition, as I just discussed. So we had this hypothesis as well. And then these clones are bad for multiple reasons, right? So one of them could develop a cancer, but also they could be affecting the function of the tissue because the tissue is just being remodeled with all these clones, but they could also be good. So it has been described that in the liver, having these clonal expansions is good because it helps regenerate the tissue. So it's still a lot to learn about this, and it's a very active field of research. Okay, so back to our lab then, we continued after having discovered all these things, trying to figure out how can we detect ovarian cancer early. And so the next study was done with the PAP test that I mentioned. And here what we found is that the tumor mutation could not be found easily. Only 3 out of 9 cases had it. It's not surprising because the sampling happens much farther. But we had still many TP53 mutations. And what this article showed us is that in this plot here, you can see in yellow, the individuals with ovarian cancer, those were the ones that seemed to have more mutation frequency. And so what we started noticing is that maybe people with cancer, we are not collecting here the cancer mutations, but other clones, and maybe they have more of those clones. And so what we are doing now is reformulating our hypothesis. So with all these liquid biopsies, we are not trying to fish the tumor mutation anymore. We are hypothesizing that maybe individuals that have cancer have more clones to start with. And when we are using these biopsies, we might be expecting more clones, larger clones, because these people are just producing more of these clones to start with, and that increases their chances of getting a cancer. This is a hypothesis, but I'll show you just some preliminary data that we just published. Taleya in my lab did another uterine lavage study, and this time we put more genes there. And the other genes didn't give us many clues. It was, again, back to TP53, and we found more mutations in individuals with cancer compared to benign. So it looks like we are finding more support for this theory. And we are just completing now this year a larger study with 284 patients. So hopefully this will set the answer for this type of approach for ovarian cancer detection. And so the summary of this part is that in terms of detecting the tumor mutation, this approach detects quite a bit. This one detects not that much, but overall, if we want to take this to the clinic, maybe the biggest value is just counting all the mutations overall, because this might be associated with having cancer. So we'll see what the studies tell us. And so with that, I think I'm just gonna move to the last part, which are clonal expansions in normal colon. Unless you have any questions, or otherwise we can take questions at the end too. Okay, let's move to this one since we are past the half hour now. So for this study, the idea is similar. What we are thinking now is that since we know normal tissue has mutations, the colon is a tissue that, as you know, develops polyps, and then the polyps go into cancer. So our question was that maybe this is a tissue that is accumulating these mutations over time, and there might be more of those in individuals with colorectal cancer. And this is very easy to check because when we have colonoscopies, individuals, so here colonoscopies just for cancer detection, individuals get a sample of their colon. And so we do have those tissues. And when the individuals have colorectal cancer and have a colon resection, we can also get the normal colon. And we were interested in normal colon 10 centimeters from the cancer here, so away from the tumor, to see if it was really normal or not. And so Julia, a grad student in my lab, did this pilot study. And what we found is that there are actually many mutations in normal colon, not surprisingly. And some of these are really pathogenic mutations. So in this case, we had to sequence KRAS. It's a very common colorectal cancer gene. And the bad mutations are in these positions. And we found 7 of those mutations in normal colon, the 7 of them in people with cancer, none of them in people without cancer. TP53 has many more mutations. We know that from other studies, and everybody carries them. So we had some in here in no cancer, but people with cancer had more. And if we put all together with this other gene that is also a colorectal cancer gene, you can see that people with cancer in general tend to have more of these mutations overall. So this supports this hypothesis. This slide is a bit busy, but shows all the data and other important points. So I'm going to walk you through this. The point here is the age of the patients. So you have here individuals without cancer and here individuals with cancer. So again, the same ones in the study that was specifically designed to have younger than 55 years of age and older. And it was matched for no cancer and cancer. The idea for this is because we know now that there is a new wave of colorectal cancer in younger people, younger than 55 or even 50 years of age. So we wanted to see how are their normal colons. And what you can see here are all the genes that we sequence. And these are obviously the patients, and each square is a mutation. You can see TP53 always has the most mutations. And you can see the darker colors as before mean larger clones or more pathogenic. So what I want you to see here is the comparison between individuals with cancer that are young and individuals without cancer that are young. And you can see that this normal colon has many more mutations, right? So for individuals younger than 55, we do see an abundance of mutations in the normal colon. And we really want to investigate this to see if we can use this to first understand why these patients get cancer so early, and then to find methods for early detection. As I said, colonoscopies are routine, and we can get these samples and then do this analysis. Another important point here, I mentioned that the polyps predisposed to colorectal cancer. So when we counted TP53 mutation frequency, it was higher in individuals with polyps as well. And so this shows a bit of the continuum of progression. So we think this is an important study to continue, and we are doing now this in collaboration with other big groups that have access to many more samples, and we have a grand, big project to get this done. And so the summary of this part is that somatic mutations occur as well in normal colon, not big surprise, but more in individuals with colorectal cancer. And we want to, well, the specific genes are these two, TP53 and KRAS. They are prevalent in individuals with colorectal cancer. And we want to use these measurements to assess colorectal cancer risk. Okay, and with that, it's good timing because I'm going to wrap it up now, trying to put all together in a couple of slides. So this is a model that I have been explaining for several years now, but I think it's in a little bit similar to the one from Peter Nowell. This is cancer evolution, but takes into consideration the full life of the individual, right? So what we know now is that evolution is not just at the cancer stage. It happens with life. So what we are representing here are cells. When we get mutations, mutations sometimes kill the cell, and that's the end of it. They are bad. Sometimes they do nothing, and so you can see them here. The cells just persist with their mutations. They are not good nor bad. But sometimes these mutations are pathogenic. They allow the cell to expand and make a clone. And these mutations, as I explained, they come from all these different sources. This is normal. This is aging. Why we don't all get cancer if this is aging and we all have these clones that are in theory predisposed to give cancer? Is because we have a lot of what we call tumor suppressor mechanisms. So the tissues are engineered, our tissues, not to allow these cells to grow beyond their boundaries. That's one. We have something that is called senescence that would make a cell die, or, well, die is apoptosis, but these senescent cells sort of stop their function if they are getting to have a lot of problems or mutations. And we have the immune system that is supposed to see all this and also make it disappear, right? So all this works very well to control cancer. However, we know that sometimes this might not work, right? So why that happens is what we need to understand. So how come these clones eventually turn into a tumor? So in the past, we used to say more mutations. So you can get a mutation in any of these systems, and then this keeps going. But now there is a lot of discussion about the environment being able to allow the expansion of these clones in some individuals and not others. So this is what we call promotion. All this is initiated. What is promoting these clones later on? That's a huge topic of research now, and a lot of people, very smart people, are just putting their brains and ideas and data into this part, trying to understand what eventually makes these clones produce a tumor. And so in terms of early detection, what we want is to be able to sample here and see either people that have a lot of these clones that might be starting, or even just sampling the clone itself. So that's what we think we can do now with these high resolution technologies. So I'm going to tell you the conclusions, and then we'll have time for questions at the end. But just to summarize, I've told you 3 stories. The first one is the basic background of somatic evolution. So hopefully it is clear to you now that cancers are an evolutionary process, but this also happens in normal tissue, is part of aging. And we can see this now because we have this resolution to see all these mutations. In terms of early ovarian cancer detection, we might be able to do some detection of the tumor with the uterine lavage, but it looks like at the end, the whole information of pathogenic TP53 mutations might be the most important to measure. And this might be not a detection method for cancer, but a risk assessment. We can use this test in the clinic and maybe have some sense that if it's high or low, then the person might have more or less risk to develop ovarian cancer. And then similarly in the colon, we could potentially access colon biopsies, check for these driver mutations and see if there are more clones and see if these patients eventually are the ones that develop colorectal cancer. So a lot of potential clinical application here. And so I started saying that we don't know much about the beginning of this process, but hopefully I've convinced you that now we can understand the beginning a little bit better. And lastly, there are many, many, many questions, right? So I've tried to boil this down into small pieces, but there is much more than we are doing in my lab. We are studying different tissues like bladder. We are doing applications in other different ways to study chronic lymphocytic lymphoma and to study how cancers develop. And so there are multiple ways that we can use these technologies. But in terms of the pending questions, some of them I have already touched on a little bit, but definitely the size of the clones is something that we can measure, and we want to measure and see how this relates to cancer progression. The good clones versus bad clones, I've mentioned that in the liver, so we don't know yet. Some clones might actually be good. And then how the microenvironment of cells contributes to clonal expansion. So I think studying the immune system and the tissue microenvironment is going to be key. And Colleen is also trying to do that in the lab now. And then these type of research also opens a lot of opportunities for interaction with other investigators in the fields of aging and exposures and cancer. So I don't think we can do this work isolated. All this is very related. We want to characterize these cancer mutations with aging much better than what we've done, similar to how Cosmic characterized tumors. So we want to characterize the normal mutations with aging. And then mutational signatures is another level of analysis of DNA based on exposure. So that's something that we can do as well, and compare individuals with increased risk of cancer versus those that do not have. And back to this diagram, now we know that there is just not one single cell here. There are multiple that are mutated, that are diverse, and these eventually might give rise to the tumor. And so with that, I think I still have 10 minutes for questions, so that's great. I want to acknowledge everybody in the lab that has done all the work. I've mentioned some of the people as I go, but this is obviously a team effort. It requires a lot of time and effort to get these samples, do the microdissection, extract the DNA, and do all the sequencing. And of course, this is done with a huge group of collaborators here at the University of Washington, and also some international collaborations. And with that, then I'll take some more questions. Thank you. Kelly.
Thanks for the great talk, Rosana. I'm wondering, with the uterine lavage test, is your aim get enough evidence to eventually make it a part of routine exams for everyone?
Yes, that's the idea. There is such a huge need for a test for ovarian cancer. The ones that have been tested and they are still used in the clinic, but they really don't offer much benefit. It's just an ultrasound essentially, and then a blood biomarker called CA125, but it hasn't led to improve survival at all. And so the need is there, it's very clear. And so there was a lot of interest when we first published this. And right now, this larger study is actually one of the collaborations here with people in Europe. The group that invented this catheter contacted many institutions to do this uterine lavage collection. And so that's the data that we will be analyzing this year. The pandemic was just a big hit and the collection stopped, but finally we hope to get this data soon. And then in parallel here in the States, there has been a lot of interest too, and there is a network called Early Detection Research Network that has started collection as well. So it's a parallel effort here from 5 or 6 different institutions that are collecting uterine lavage. So the need is huge, and we are going to need now all the brain power and everything that we can to extract the data out of this, because unfortunately having so many mutations in TP53 makes it much challenging, right? We wish there was not that much biological background, but hopefully we can still see the signature. Yeah, so eventually it will be used, I hope. Yeah.
My question is very silly, but I'm wondering, you see all these TP53 mutations that are maybe not pathogenic, but they are in patients with and without cancer.
Mm-hmm.
Did you also see those with the sample of skin? This be all around the body, and I'm asking this because I'm wondering if you could use that to filter mutations away, so then you can focus on the ones that are dead.
Yeah, that's a great idea. There are no silly questions at all, by the way. That's the sort of conversations that we have in the lab all the time. All of those are good, and a lot is unknown about these mutations, right? We've had this same idea sequencing the blood, at least to start with. So yeah, just get a control tissue, right? And see, okay, is this individual carrying more of these mutations overall? Ideally, it would be 2 or 3 tissues, right? And why that could be, who knows, but at least you can control a little bit. So we've done that. The blood is complicated because it has its own dynamics of clonal expansions, right? So at least what we've been able to do is filter out if a clone in blood has been found in the tissue. And that has happened, right? So you can see some of the tissue, if it has blood vessels, obviously, you might see clones there, the chip clones. So yeah, we use chip clone filtering in the data to at least get rid of whatever is in blood. But it would be great to do multi-tissue analysis in many patients to try to understand a little bit better what are the levels and do they compare? Does an individual have high levels in all the tissues or not? We do not know yet. There is very little that is known. Yeah. Question over there.
Insurance companies pay this expense. Is this a real problem that you face and that you perceive that you have to tackle in the
That's an excellent question, and you are totally right. Yes, sometimes that could be a struggle, even here in the most basic tests that get done routinely. So the University of Washington has, in my department actually, which is lab medicine and pathology, that's where this testing gets done in lab medicine. And there are several tests for clinical indications. The BRCA genes and others are all together in what we call the BRCA test. And we have the ColoSeq test and many others that are being developed. And my understanding is that the insurance companies for many patients would pay, but depending on the indications, there might be issues, right? And so that's the role for us as clinicians. I'm not a clinician, but for my colleagues that are clinicians and as scientists as well, because that's how we collectively get data to try to understand a little bit better. Now, if we do prove the value, right? So I think that's the importance. If we do prove the value of using uterine lavage as a risk assessment, I would hope that this is then approved by the insurance as a similar to like a cholesterol test, right? Like something that you need to tell you if you are at risk of cardiovascular disease or not. So this would be something similar. It is true that they are expensive tests. And so that's where the problem is. But the sequencing technologies in general are becoming much, much cheaper. So that's the other hope here, is that it's not nearly as expensive these days than it used to be. And when people think, my colleagues that are clinicians, about is this feasible in the clinic, the main consideration is cost, right? So it's not, can we sequence with these fancy technologies? Can we do this under $500? So if we can, then we'll move it to the clinic. But if we cannot, then that's a no-go, essentially. Yeah, great question. Yeah. That's a really good question, and that relates to a slightly different topic related to liquid biopsies. So if you had to do screening for cancer, this is still not happening, right? But if it could be done with a blood sample, some companies have started trying to market this idea, like you have a blood drop and you can extract DNA and then see if there is cancer or not. So I don't think the technology is there yet, but the point here is that in the blood, you do have circulating tumor DNA if there is a tumor. So tumors shed DNA, all cells do, but tumors as well, into the bloodstream. That DNA is called circulating free DNA, but there is a subset of that that would be circulating tumor DNA if the person had a tumor. That's doable. That happens all the time. People are doing ctDNA a lot, so that's not science fiction. But the idea, and actually I don't know the field enough, I don't know what stage it is, but the idea here is that if you sequence that DNA, then you can identify the tissue of origin, as the question before, right? Because there are certain features in this circulating tumor DNA that might inform that this might come from a tumor in the liver or the brain or the pancreas, right? So that's in theory doable because there are some features in the cell-free DNA that both allow you to do that. But I don't know at what stage this is. Circulating tumor DNA is used these days a lot for monitoring disease. So the patient had cancer, got a treatment, and now can you see over time if the cancer is gone? Definitely, that's the best application. But early detection, in theory, the same principle applies, right? You could just try to find these tumor mutations, but I don't think we are there yet. The last studies that I saw, and that was not recently, they were trying to add into the mixture protein, not just the cell-free DNA, because it was not very clear if this was doable. Yeah, does it answer the question? Yeah. Jacqueline.
A lot about somatic evolution and clonal expansion. I'm wondering if you could use duplex sequencing to detect a mutation, or I guess two mutations in one cell, where it's like both allelic copies are mutated only in one cell, but not necessarily the expansion of this.
Yeah, so single cell duplex sequencing, that would be the notion. It's something that we would love to do. So when we do this, we are just getting all the cells, extracting all the DNA, and that's a soup of DNA, and then we sequence. And we don't know, we might see multiple mutations, but we don't know if they came from the same cell or not. We would love to do that, and it's doable. I think it's technologically doable. There are many methods these days about single cell, everything. It's just a question of making it work. So I think we'll see that one day. The type of questions to answer are a bit different, but definitely there are many questions that could be answered with that technology. Yeah, so I hope we'll see that. That would help, for instance, in the lavage, right? If sometimes we say, well, instead of finding one mutation, find multiple, right? That's the strategy for the circulating tumor DNA. One might not tell you for sure, but if you have multiple, then you get much more certainty. Any last question? One more? Yeah. Hi.
So you're looking at the individuals cancer who are young, colorectal cancer, who are young, and you're detecting mutations that are in the normal colon. What is promoting the expansion of these clones in the normal colon? But these individuals do have colorectal.
Yeah, great question. No idea. So there have been now many epidemiological studies about this subset of individuals that are getting colorectal cancer at young age. And so, of course, this happened only 2 or 3 decades ago. So it's kind of a new epidemiological finding. And so, of course, all the hypotheses are the typical ones: diet, environment, but it's unclear what is driving these clones to expand. But it's a really great model. I think I was very driven to do this study in colorectal cancer because I think it's a really good model to study these pre-existing clones and then try to figure out methods for their expansion, what could be happening. I don't think it's mutations necessarily. I think it's environmental, but that research has to be done. And as I said, there are collaborators with a lot of samples and expertise working on that. The microbiome is another big one. So one of these collaborators is trying to study these precancerous lesions with mouse models, manipulating the microbiome. And I think that's very interesting too. Well, we are past the time. So thank you so much for staying, for being here, and hopefully you learn. Thanks.
Thank you